turn over. I like your shoes too.
Thank you. I bought them in Montreal. They are strong, warm leather work
They
shoes.
look comfortable.
Yeah, they're unbelievable. They're 6 years old. You would never know it. I walk to work from Coogee every day. They're amazing.
Just up the hill.
Just up the hill when you're late, which I always am. It's actually keeps you fit.
Why would you like to start? Do you want to tell me about something about the Children's Cancer Institute? Or
I can tell you about. Am I looking at you or the camera?
me?
Yeah. I can tell you about Children's Cancer Institute. So we are the only dedicated children's cancer research facility in the country. We are the largest children's cancer research facility in the Southern Hemisphere. I was the first postdoctoral scientist who was appointed here back in 1984 when the doors of the laboratories first opened, and it was established by parents and doctors of children with cancer to do research that would make a difference for kids. So from the very beginning, it was very outcomes focused. At the time, it was the first time that there'd been dedicated laboratories for children's cancer research in this country, because only 10 or 15 years prior to that, all children died of cancer. When I was a child in the 1950s, if you were diagnosed with cancer, you died, basically. And then with the advent of chemotherapy, which was pioneered in children with leukemia, and then combination chemotherapy, which was pioneered in children with leukemia, and then multidisciplinary cancer care, which was chemotherapy, radiotherapy, surgery, all of which we take for granted now. All of that was pioneered in children with cancer. There was this rapid increase in survival rate for kids. And by the time I got into this field in the early 1980s, which was when these funds were raised to open the first lab, the survival rate had raced up to about 50% and then gradually to 60% and to then 70%. It's slowed down and plateaued since then. That's another story. But it really has been one of the great miracles of modern medicine that we've gone from 0 children surviving a disease to where we are now, which is better than 80%. And so Children's Cancer Institute was in that from the very beginning. And I've been part of that journey for 42 years. It's been amazing.
This is a different sort of cancer, or not a different sort of cancer. How is it explained to me? Children react differently to adults in some cases. I can't make a generalisation. I have to let you do that for me.
That's okay. Children get different types of cancers to adults. So most adult cancers, as we understand them, have some sort of environmental cause. So if you're in the sun too much, you get skin cancer. If you smoke too much, you get lung cancer. If you eat pickled foods, you get bowel cancer. There's a whole range of known carcinogens that drive adult cancers. Children have none of those. They're pretty much either born with cancer or they develop cancer in the very early years of childhood. And what they are diseases, what we say cancers of embryonal origin. So they come from the cells in the embryo that go to form the tissues in the fetus and then in the baby. So the commonest childhood cancer in children are blood cancers or leukemias. And so that's a cancer of the cells, from the primitive cells that are making the blood-forming system. Something goes wrong and they don't develop into those normal cells and they get stopped or arrested at that point in their development. What you get is this very primitive cell that has lost all its normal growth controls but can go on and divide and multiply in an unlimited way. And so you get blood cancers, you get bone cancers, you get muscle cancers, you get a nervous system cancers. So cancers that are simply very rare or non-existent in adults and from a different origin, not from these chemical causes so much or exposure causes, but more from developmental causes. So they're different types of cancers. And so they need different types of treatment. We need to do different research. We understand that the genetic drivers of cancer, now that we are able to molecularly dissect what's going on in tumours, there's only about 50% of these molecular drivers that are common between adult and childhood cancers. Fully half of them are completely unique to children's cancers, which is why you need a dedicated children's cancer research facility like this.
It sort of explodes. Well, is it? Let me get the question right first. As you grow, of course, everything's happening with your body, and is that going to affect your cells if you're a kid? That wouldn't affect adults because you're going through the growth spurts and everything else. Is that one of the reasons why it's so different?
It's one of the reasons that contributes to why a childhood cancer journey is different to an adult cancer journey. Children's bodies are growing very rapidly. And so the cells in a child's body, by their nature, are dividing and multiplying very rapidly. That actually has some advantages in treating cancers because sort of counterintuitively, it's easier to kill a rapidly dividing cell than it is to kill a slowly dividing cell. The earliest of the chemotherapy drugs were drugs which were simply designed to kill rapidly dividing cells. So they targeted the replication systems in the cell or the cell division because that was happening much more rapidly in cancer cells than in normal cells. There's a corollary of that, though, which is that because the normal cells in a child's body are growing fast, many of these anti-cancer drugs have terrible side effects for the children because they're damaging the normal, rapidly growing cells in the child's body. So one of the things we learned about anti-cancer treatment very early in the 1970s or so, after this first generation of children whose lives had been saved through these chemotherapy drugs, they were left with really dreadful side effects. They had cardiac damage, they had metabolic damage, they had fertility disorders, they had intellectual disorders, they had growth disorders. You know, there was a generation of severely damaged children. In many cases, these effects were not only, lifelong health complications, but in some cases, fatal complications for these kids. And even today, between a half and 2/3 of all children, childhood cancer survivors, their journey isn't over if they're cured. They still have these health challenges as a result of the treatments that were given to save their lives. So that's a particular... Survivorship is a big issue with both adult and childhood cancer, but it was recognised in children with cancer, and it's a bigger problem in childhood cancer survivors than adults. So our research is not only about cure, but it's about quality of life and the type of journey that these kids and their families have to go through, which even today is still horrendous for families. Whether the kids live or die, they all tell you it is the most terrible time in their lives.
And that's not the cancer lying dormant.
Sorry, can I just have a little bit more?
Listening to starboard?
Indeed. That should happen after 5.
Yeah.
We can help with that.
So look, it's not, I guess the point I was making, it's not about the cancer remaining dormant and coming back. You're actually saying a lot of the survivorship has been damaged by the treatments.
There are cancer cells that escape treatment. So you treat, you get the child into full remission, and you think they're cured. But sometimes there are small numbers of surviving cancer cells, and this is every parent's nightmare, that at some point after your child has been well and has gone back to school and you think everything's okay, suddenly the child will relapse. And what that's due to is that there are small numbers of surviving cancer cells that have escaped, have hidden somewhere in the body, and then will start dividing and multiplying again and come back. And once the child relapses, you've already given them all your best treatments, all your best drugs. Once they relapse, their chances of surviving long term are much, much lower. So a lot of our research is focused on how can we identify children at high risk of relapse because they've got what we call this minimum residual disease somewhere in their body. How can we predict that? How can we identify it? And how can we intervene before the children relapse to save their lives. And we're very good at doing that here. We've developed a number of molecular tests that allow us to predict which children are at risk of relapse, and we intervene and give them therapies that avoid that relapse, and therefore they're alive and well years later, whereas we know that they would otherwise have died.
It's a massive game of hide and seek, is it?
Mm.
Could you say that?
Tumours are terrifyingly smart if you want to give them human characteristics. They adapt, they mutate, they hide, they avoid the immune system of the body, which is actually designed to recognise something that's abnormal and to attack and kill it. They actually are capable of changing the environment around them so that they can literally disguise themselves from the immune system. It sounds nuts, but that's actually what they do. And so a lot of our newer immunotherapy is working out how to drop that mask from the cancer cells so that the immune system can come in and attack it. It is a game of hide and seek. It's a game of whack-a-mole, if you like. You know, you're hitting one, you come in with a drug, you treat it, you kill 90% of the cells, but there's 10% that mutate and respond and change, and then the tumour comes back in a different way and you whack it again. And it's a journey to try and find ways of curing these children. I actually sometimes find it a miracle that we have got to the point that we are curing 85% of these kids. We're now doing it smarter, better. We have personalised medicine. We can harness the genomic revolution to look at the specific genetic makeup of each child's cancer and tailor the treatment for each child to the particular genetic drivers in their drugs. We lead Australia's National Child Cancer Personalised Medicine Program. We've developed that here. I think it's the best program in the world. It's generally regarded as that. We're very proud of it. And those sorts of innovations are giving hope to families, changing the game. But it's a process of attrition. I think we will get there. I have got no doubt if we can go from 0 to 85% in 60 years, then with research, with funding, with the best scientists, with international collaboration with clinicians, we'll go from where we are currently, where we have 3 kids a week in Australia still dying from cancer. We'll take that from 3 to 2 to 1, and ultimately we'll have 0 children dying of cancer. I absolutely believe that.
In our lifetime, going from everybody dies to 85% don't.
Pretty good, eh?
Bloody oath.
It really is
I mean,
amazing.
no one, most of the people seeing this, unless they're older, but younger people like ourselves, would have no memory of, what would you say, the actual despair?
Look, if you speak to people of our more senior generation, there are many who recollect children in their classrooms who simply stopped coming to school one day and quietly disappeared. It's amazing how many people tell those stories. Or my sister died of leukemia and they just got sick and died because there was no treatment for it. Prior to the advent of chemotherapy in the 1940s and 50s, the only way of treating cancers were you either cut it out, surgery, or you burnt it out with radiation. And historically, that had been what was done with cancers. That was all we knew about them. What we also knew is that didn't, unless it was a particular small localized tumour, it actually didn't work because cancer doesn't kill you from the primary tumour. The cells spread through the body and attack the bone and the brain and the liver and all the other tissues. It metastasizes or spreads, and that's what kills you. And of course, neither surgery or radiotherapy impacted that. It was the discovery that you could take drugs that could systemically treat the cancer that revolutionised our field. And although children's cancer is less than 1% of the entire cancer burden, it's a huge differential between the number of adult and paediatric cancers. In fact, almost every innovation in cancer research and care has been pioneered in kids. So I mentioned chemotherapy, combination chemotherapy, multidisciplinary cancer care, survivorship, psychosocial support, precision medicine, molecular testing, CAR T cell therapy. All of these have been pioneered in children with cancer. And it's because there is a global research effort. The numbers are small. Every child's experience counts. So the idea of multi-institutional and multinational clinical trials to drive the field forward, that came from pediatric cancer as well. And it has done that. It has driven the field forward. And it is remarkable that we've gone from a time where every child died to now most families knowing that if their child has a cancer diagnosis, there is a good likelihood that their child will survive. They will still have many challenges. The family will have many challenges. As I said, the journey is hard, painful. Surgeries, interventions, lumbar punctures, anesthetics, scans, missed years of schooling, challenges with young kids growing up and not fitting into school because they've been away for so long, siblings going through guilt. Why was it not them that was suffering when their brother or sister that they love is going through this terrible thing? It's an incredibly destructive disease still. We have so much more to do till we solve this. That said, it is truly a miracle of science and modern medicine that's been achieved in our lifetime.
Yeah, because DNA was what, 53?
53. When I started on my journey in cancer research, we used to look down the microscope at cancer cells, and we could see that they looked different from normal cells. The chromosomes looked a bit weird. First of all, there wasn't the right number of chromosomes in some cases, that there were more or less than there should be. But the banding patterns of the chromosomes weren't normal. There were extra bits and there were missing bits. And it was like, this is a mess, but what's it telling us? And all we knew was that they looked different. Fast forward to the genomic revolution and our ability to sequence the human genome, which took 15 years and however many billions of dollars to do. That has revolutionized what we can now do. I started studying cancer cells gene by gene. That's a bad gene. I'm going to find out whether there's lots of that gene in a cancer cell, and I'm going to find a way of switching it off. There's 30,000 of these genes. That's a pretty slow way of doing it, but that was all we had at the time. Now, as part of our personalised medicine program, we sequence the entire genome of a child at diagnosis and use that information 4 weeks later to make a personalised treatment recommendation. That is now standard of care. Who would have thought that would have been possible? And so we're not looking at one gene. We're looking at the entire genome. We're looking for every rearrangement. We now understand what those funny missing bits and pieces were. We understand that there is what we call genomic instability in cancers, that one of their hallmarks is that chromosomes break and rearrange. And when they do that, you get a bit of a gene over here that's designed to say, make the cell grow, make the cell grow, make the cell grow. And normally it would have been attached to a proper growth protein that was involved in that cell growing. You put that in front of a bad actor that then starts making a protein that turns that cell crazy. That combination of those two coming together is literally fatal, because you've now unleashed a signal that will make these tumour cells unstoppable, basically. But we understand it now. So we can now design drugs that interfere with that new interaction coming together and saying, I know you want to do that, but we're now going to target every cell that's got that, because we can find a bit here and a bit here. And the only cells in the body that will have tab A and slot B are the cancer cells. So we can design targeted drugs that will only hit the cancer cells and not the normal cells, because that rearrangement doesn't occur in any of the normal cells. That's what we mean by targeted therapy. This has been so new. This is in the last 20 years or so, it was a dream at the time we started. It was a dream that we would one day understand what these crazy chromosomes were. Now we do. We understand the scope of the challenge. We haven't got all the solutions yet, but at least we understand what we're dealing with.
Okay, my head's spinning, and it's about 3 things I want to leap in there and talk about on the way through. Wait, what was the first drug?
Methotrexate.
Sorry, just Tom, just from the aircraft.
Oh, here comes one now.
Wow, you've got sensitive equipment.
He's
Yeah.
a sensitive man.
Okay, brilliant.
The first drug was a drug called aminopterin. It was used in children with leukemia. It was used by a doctor in Philadelphia called Sidney Farber, who was actually subsequently disciplined for poisoning children because he was told that all of these children were destined to die. So what the hell was he doing giving poisons to children? He should be leaving them alone to die in peace. His poisons demonstrated that leukemia cells had because they were growing so rapidly, they had a requirement for a particular growth molecule in the cell called folate. It was just needed to make DNA for the cell to divide, grow, and multiply. And aminopterin was an antifolate. It blocked the synthesis by the cell of these folates. And the leukemia cells were incredibly dependent on that. And when you basically switched off the supply of their growth protein, the cells stopped dividing. So his first discovery in the 1940s was the demonstration that you could stop for the first time with a drug the growth and proliferation of tumour cells. And he was able to put children with leukemia into remission. And by remission, what we meant was that if these cells grow in the bone marrow, you can put a needle into a bone and suck out some bone marrow, and you can look at it down a microscope. And in a normal bone marrow, all you'll see are the cells in the marrow. They're part of the blood system. They're part of the infection-fighting system. They're part of the system that makes blood cells clot if you you cut yourself. That's all you should see. In a leukemia patient, you do that. You stick a needle in, do a biopsy, and you look down the microscope. It is 100% replaced with these primitive leukemic, we call them blast cells. So cells that should have gone on to make all of these infection-fighting drugs and clotting drugs and all of the others. None of them are present. These leukemia cells are early primitive blast cells that, as I said, have got stuck in their differentiation. They've never made it there. They have a growth advantage, and they colonize the bone marrow and they overgrow the normal cells. And so when the child starts looking pale and starts bruising and starts getting coughs and colds and is generally unwell, it's because all of those cells that are meant to deal with that have been wiped out. And all that's left is this bone marrow chock full of leukemia cells. And the inevitable outcome of that is that 8 weeks later, that child is dead. And that's what used to happen. That's why these children, I've heard them referred to as white ghosts or pale ghosts, because they were pale. And then they just faded away and died. He showed that you could clear the marrow. Can you imagine what that discovery would have been like? As a researcher, as a
When was
doctor,
this, like late 40s?
late 40s, he showed that you could, if you gave these drugs, single drug, aminopterin, and then you looked in the marrow, it would be clear. The problem was every one of those children relapsed. So the disease came back, and it was like, why did that happen?
But it was a clue, wasn't it? So we got
It was a signpost. It was like, that's the path. How do we do this better? From that came the idea, well, if that's one pathway that the cells are dependent on, they must be able to escape that and find another pathway. So let's put another drug in there. And that led to the idea of combination chemotherapy. And now standard treatment for a child that's diagnosed with this disease is it still includes methotrexate, which is a slightly modified version of what aminopterin is. So still, 80 years on, we're still giving almost effectively that same drug to children. But in addition, we're giving a cocktail of other drugs that block other aspects of the replication of that cell. So they can't divide. And what that did was it prolonged the remission, and suddenly a year later or 2 years later, as they began to add in radiation. And the reason for the radiation was that they found that there were sites in the body that the cancer cells could hide. And that was in the spinal cord or in the testes of young boys. They're sort of sanctuary sites, as we call them, and the tumour cells would hide there. So you had to irradiate. The problem with irradiating the brain was another site. Then these children got intellectual deficits. So it was a learning process. But through all of that, the first generation of survivors came through. So long answer to your short question, what was the first drug? That's what it was. And every cancer researcher knows the answer to that, or should.
It's a bloody breakthrough. It's wonderful.
It was extraordinary. It was extraordinary. And I have to say, I gave a talk a couple of weeks ago. I was asked to reflect on the history of cancer research, and I gave the heading to the talk, Big Ideas from Little People. And in fact, that's what the history of childhood cancer research has been. There have been these breakthroughs that have revolutionised each of those things. Oh my God, a drug can do this. A combination of drugs, can you do that? Oh, you add in radiation, that'll protect them. But look what it's doing. Okay, so now we have to back the doses off. Then we have to find ways of predicting who are the kids that are really high risk so that we can give them the more intense treatment, but not the others. How do we do predictive testing? It's just, and the other is predisposition to cancer. Why do these children get cancer in the first place? They're not drinking or smoking or partying in the sun when they get diagnosed with cancer at 6 months of age or 12 months of age, or even when they're born. We've shown that if we analyse the normal genomes of these children, so we look at the DNA, not in the tumour cells, but in the normal cells, in 1 in 6 of these high-risk cancer kids, they have a change in a gene that we know gives a susceptibility to developing cancer. So it is a combination of genetics as well, because that will be heritable, those changes. That means that it's come from a parent. It's likely to be in a sibling. The more we understand about this, the more we can identify who's likely to be at risk of getting cancer, and therefore we can start looking at prevention. And to me, that's the holy grail, that one day we're not treating these kids, we're stopping them from getting cancer in the first place. That's another huge, exciting area.
One that Isaac Asimov certainly didn't see coming, or most of the science fiction writers, which is interesting.
It is interesting. We were reflecting on AI the other day, and boy, he saw that coming. It's just when I read it, I read iRobot, I read the Foundation series, I read every single Asimov book that was ever read. He was pretty prescient. It is remarkable.
We got a great interview with him from about 1978 or something.
Wow.
Predicting the future.
Yeah.
Okay. And it comes up cancer. He said, ever, maybe?
Yeah, it's not ever maybe. The green badge that I took off our logo at Children's Cancer Institute, it's not if, it's when. We absolutely are driven by that belief, which is that one day we will cure childhood cancer. We may make it a chronic disease. I've got an autoimmune disease. It's an autoimmune disease that 40 years ago killed every woman that got it. I now take my 6 pills every day, and here I am, alive, well, minimal side effects. I think.
Swimming with whales.
And swimming with whales. And, loving what I do every day. I think that's where we will get to with some of these kids. You see some of them where you find the genetic driver of their cancer. You can see that they're in intensive care, they're on life support, this cancer is killing them. And then you sequence their DNA, you put them through our Zero Childhood Cancer National Precision Medicine Program. You find the genetic cause, and if there's a drug that targets that particular genetic change, it's miraculous. It's just miraculous. The tumour just melts away. And we've got examples, not every child, not nearly every child yet, but we've got the walking, talking, living, growing, breathing examples of kids who this happened to now 5 or 10 years ago. And they're growing up into teenagers and they were destined to die. So it's pretty special to be in this area.
We haven't concentrated too much in the documentary on chemo. We're sort of going through the timeline and say, oh, it came from mustard gas and stuff in World War One, which is,
Yep.
I think would surprise most people. You're going to have to reword this for me because my voice is never going to be on the documentary.
That's okay.
Is it of limited use? It takes you so far, doesn't it?
Chemotherapy is still a mainstay of cancer treatment, both in adults and children. It is the way, it was designed in many cases from compounds that were known to be able to stop rapidly dividing cells. One example is from mustard gas, which was designed to bind to the DNA and damage it. That's what its job was. Now, if you have a cell that's dividing more rapidly than another cell, you can give it a drug like mustard gas. We call them alkylating agents now. And the successors of mustard gas are now drugs like cyclophosphamide, which have been now treating cancer patients since those earliest days. They're old drugs. They're still the mainstream part of treatment. I do look to a future where either their doses are highly reduced so that the side effects from them are reduced and we partner them with more of these targeted drugs, with immunotherapy, with CAR T cells, so that upfront the damage that is done is better. We haven't yet got the technologies to replace it entirely. So we live with it, we manage the side effects from it. When I say we, this is a cancer research laboratory. I'm talking about the clinicians who treat the kids. It's a collaborative effort. You can see the space we're in now, clinicians and scientists all in one facility, because that is the future, this partnership. But
it's about targeting. If you can target that and deliver that drug
Yep.
in lesser amounts directly to the core of the
Yep.
tumour.
And with a treatment that is specific to the cancer cells. And there's more and more work that is being done on that now. There are targeted antibodies that hone into the tumour and then kill only cells that express a particular protein that is on the surface of cancer cells and not normal cells. CAR T cells are designed to re-engineer the normal infection-fighting cells of the body to attack only the tumour cells when they're reinfused back into the child's body. There are these targeted drugs that are not specific to individuals, but are specific to any of these particular genetic rearrangements that we see. If that rearrangement is only present in cancer cells and you can make a drug that will attack that, that's going to be an effective drug because it's less likely to have side effects. So there's a whole new generation now of people working on these more targeted, safer, more effective therapies that spare the normal tissues and give better quality of life.
You got me going earlier with chromosomes.
Mhm.
Just to be I wasn't a scaremonger, somebody else was when we came to chromosome 2.
Right.
Because they said this has shown signs of being genetically engineered 200,000 or 300,000 years ago. And I went, how the hell can you say that? He said, you can see where the telomeres match and how they joined. So that could happen in evolution or nature. I'm just playing around at the moment, but
Yep.
It's like, have you heard anything about that?
So we are increasingly now looking at historical events that you can map to, how do I describe this? There are many things in the human genome that we look at and we actually have no idea why the DNA looks like that or what it's doing. Only a small fraction of the DNA in our tumour cells actually makes proteins in any of our cells, not just our tumour cells, but any of our normal cells in the body. Only a small portion of them are active and make proteins that are useful to the cell's normal activity. There are vast tracts of the genetic material in our normal cells that we call either the dark matter or the silent genome, stuff that's there and we think, what's it doing? So now there are teams, including teams here at the Institute, that are mining that data and that information because it ain't there for no reason. Nature isn't like that. It does things for a reason or things have happened that changed it. The telomeres one I won't talk about because
Oh, no, this is pure science fiction. Whoever's doing it.
Yep.
Okay, that's fine.
But I'll tell you one that we are working on, and that is we've known for years, decades, that in the cell there are, in the DNA, in this dark matter, in the stuff that the useless silent DNA, there are small repeat sequences, just little random cassettes of genetic material inserted randomly through the genome. We've called them lines or signs or alloelements. They've got all sorts of different names. There are hundreds of thousands of these copies. What are they doing there? So we have been investigating those. The critical thing is that they also are capable of moving around the genome. If you actually look where one was at one point in time, you can see it. So we call them transposable elements or retrotransposons. They've got various names. And we've seen them. We haven't known what they were doing. We now have a major project working on these because our theory is that maybe when these elements move around the genome, they insert into genes at critical points, and maybe that's responsible for why you get this genomic instability. Maybe that's why chromosomes break and rearrange in different ways, because one of these elements comes and wiggles its way into the DNA, and in so doing it breaks that sequence of DNA, and the bits are looking for their ends, and then they rejoin with something else that they shouldn't. It sounds like science fiction, but there is clear evidence that where these elements are now coming from, and they are the product of historic, antique, ancient viruses that had integrated into the cell, made copies of themselves that allowed themselves to insert back into the DNA and have sat there. Most of them are silent. Most of them don't do anything. But some of them have this ability to make copies of themselves and to reinsert. They're made by a class of viruses known as retroviruses. And the reason that's important is that there are current real live retroviruses like the AIDS viruses. And there are drugs that block the action of them. What if we use antiretroviral therapy to stop the replication and the insertion of these random elements through the cell? Will that reduce the likelihood of cancer developing? And will that make a tumour more responsive to therapy? We're showing that that's exactly what the situation is. So we're now working with infectious diseases specialists to see whether we can use these drugs to treat cancer. We've published the first papers on it. And the reason that that's relevant is that these integration events into the genome happened tens of thousands of years ago. And in fact, the explosion, if you look in the species of the presence of these elements across the phylogenetic scale, the massive increase in the number of these corresponded to the massive increase in the explosion of mammalian species around the time when we went from the most primitive of creatures to so many other types of creatures. Is it possible that this explosion in genetic diversity came from an explosion of the integration of these elements into ancient genomes, and they're playing a part in cancer today and may give us a clue to how to treat cancer in a different way? We've just had funding for a major grant to look at this. These are ideas that would have seemed either madness or simply impossible to prosecute. But we now have the tools to look at this, to analyse every cell in great detail, to see what's happening and to design drugs that change it. It's an exciting, exciting time to be in cancer research.
So it's not just aliens then.
It's not just aliens. It is alien viruses that have no place in our genomes. And when they misbehave, it's pretty clear that they can cause damage, but they're not little green men of the type that we would historically have considered them.
It is the missing link, though, isn't it? Well, once you get, if you talk about those chromosomes, when the apes went one way and we went another, that's why it happened, how it happened. Too much radiation, who knows?
Who knows? But maybe this is all part of how these developmental or these genetic changes that have predisposed to cancer development have come about. And maybe it'll give us a clue to treating them. And our ability to look at these questions is being made available by new technologies, genomic, proteomic, looking at the genes, looking at the proteins, looking at the way the genes are modified as they make proteins. There are so many things that we can do now that allow us to begin to address the cancer problem in an almost surgical precision way, whereas before the old chemotherapy drugs were like a blunt instrument. You just threw them at cells in the hope that you would stop their growth, and their collateral damage was massive. We're getting so much better at that. Not a solved problem, such a long way to go, but it's just tantalising how much closer that future is getting.
And 20 years. I mean, that's what blows. I mean, it's funny talking to Lauren Brown and a few other folks.
Yeah.
Oh, it's really old technology. I said, it's 20 years, Lauren.
Yeah,
It's not a long time.
yeah. The
Perspective is interesting. Isn't
The
it?
pace of change of technology is faster than anything I've seen in the last, in the first 10 or 20 years. The innovations were remarkable, but they were slow, fewer, and further between. Now there's new techniques every week. There's new approaches, there's new tools, there's new findings, there's new clinical trials. There's a real sense of momentum and excitement, I think, in our field.
I was talking to Philip Procan, and he was like the same as you when he was, I do one at a time. I know this machine.
Phil
gives
Robinson.
us- gives us like, I think they've got 36,000.
Yeah,
just
that's
to
right.
get in the library.
Yeah.
Which is fantastic. I mean, it's like, my goodness, but he goes, I had to do it one at a time. too."
Yeah,
So-
yeah, we did. We absolutely did. And you thought you were really clever if you identified the role of a particular gene. And only now you realise, goodness, what an ambitious goal that by stopping one protein, and in some cases we still can. If that protein that's driving the cancer cells is one of these bad guys, we know we can stop the tumour. The tumour still often wins because if it mutates and changes, so instead of that, it changes its sequence, so the binding site looks like that, the drug won't work anymore. And that's how the tumours become resistant. They mutate and change. So then you need second generation or third generation drugs that will be capable of attacking the tumour cell as it changes. The tumour cells are smart. As I say, they keep mutating. So it's a constant battle to be one step ahead of the game. When we win that game, that'll be the answer, not until.
Can you simply explain CAR T cells?
I'll do my best.
Well, it's going to be better than mine. Because after 6 weeks, I'm an expert, clearly.
Yeah. So CAR T cells are where you take the T cells, which are part of the infection fighting or recognition
T
cells.
maybe, yeah.
So the way in which the body is designed to fight infection or strange things that shouldn't be in the body is by having an immune system. It has a suite of cells that are exquisitely designed to do just that job with tremendous diversity. So the reason that there may be 10,000 different viruses and the body can mount an immune response against each one of them is because the immune system has been designed to give tremendous diversity. Genes have been designed to, in a very precise way, to rearrange themselves so that each time they come together, so this isn't a random process, it's a very prescribed process in normal cells, they've been designed to go through a process of molecular rearrangement so that each time they rearrange, they're slightly different. So they will be more suited to recognizing that type of germ or that type of virus or that. So this is tremendous diversity. So these cells are part of the, T cells are part of the immune system recognize something odd and fight it repertoire.
It's their response, is it? That's
Yep.
the body's response.
Yep.
Send in the T boys.
Yeah, we can take the T cells from a patient. We can grow them in vast incubators in the laboratory and genetically engineer them so that we tell those T cells what target we want them to attack. So we basically attach a sort of a special part of the molecule onto those T cells that says, go kill cells that have got that bit on it. And that bit is designed to be something that is present on tumour cells and not on normal cells. And when you've made that genetic change, so those recombinant T cells, which is what the R in CAR T cells is talking about, you make them in vast vats and then you reinfuse them back into the patient. It's the patient's T cells, so the body doesn't have a problem with them coming in. But now they're weapons because they are designed to attack. They're looking for cells that have got that particular marker on them, and they will then attack and target that.
God, you just put me in mind of the first Pope calling a first crusade.
Yeah,
Run
well,
out, everyone, off you go, lads. And just we're looking
it's
for
exactly what it does. So the first proof of this was in a child in 2000 and I think it was about 14 or... I know. It was a girl called Emily Whitehead at Philadelphia, Children's Hospital of Philadelphia, where she had multiply relapsed from leukemia, and she was on a journey to nowhere. It was pretty clear that she was going to die. And this therapy was being developed by a team of scientists at Philadelphia, and they decided to trial this because there ain't nothing else that we're going to save that child. What they found was that Emily nearly died because what the body did, this activated an enormous immune response. The body realized that there was something hectic going on. There was this army of T cells suddenly invading the body, and it set off all sorts of alarm signals. And there was what we now know we call CRS, or cytokine release syndrome, which is a hallmark of immune activation. All of these immune molecules are released very suddenly in the body. The fever goes up. Child gets very sick. The bottom line is if you don't stop that, the child will die. The brain will cook, basically. You're overheating. You've got these fevers. It is literally a fatal complication. By sheer chance, they were able to characterize what the main one of these cytokine, these sort of immune response proteins was. And they had a drug that tocilizumab was what the name was. It was capable of switching off that particular sort of immune response. It just targeted the particular cytokine that was driving.
Did they have
the-
the drug when they went in, or just like somebody went,
this-
hang on, this might work?
This was serendipity. To cut a long story short, they saved that child's life, and Emily Whitehead has become the face of CAR T-cell therapy, because I think she's 11 years on now. She's a 20-year-old, I think she is. And she's this beautiful young woman who is absolutely cancer-free. And this was the first proof of principle that CAR T-cells could work.
This is the basis of immunotherapy, is it?
It's the basis of CAR T-cell therapy, which is a type of immunotherapy. There are many types of immune therapy which harness aspects of the immune system. One of them is CAR T-cell therapy. There are others that are antibodies that attack particular cell surface antigens. There are many others, which I won't go into for the moment. But there's a whole new emerging field of immunotherapy. But CAR T-cells, this was the first proof. That was then literally approved within 3 or 4 years by the FDA as an approved treatment. So again, incredibly rapid development from first use to FDA approval.
2014 to COVID.
Indeed. And that is now very much standard of care for many blood cancers, adult as well as pediatric, lymphomas, leukemias, other types of cancers. So it's revolutionized the field of blood cancers, or hematological malignancies as we call them. The holy grail is, how do we do this for solid tumours, which is, in children, it's about 25 to 30% leukemias and 70% solid tumours. So that's tumours of organs or of the brain. So there's now a huge international effort finding better, different ways of engineering the CARs, those recombinant T cells, ways that different targets. So the first one that was targeted was a protein called CD19. Now they're looking at other targets, which are the best ones. Some are present in different types of tumours, some are present in several types of tumours. An entire suite of laboratories across the world looking to improve CAR T-cell therapy. And that then brings you to the question of, how do you know which one's best? How do we accelerate this field? And then you come back and look at, again, children's cancer leading the field. There are now international partnerships. There's one called the LifeArc CAR T-cell Accelerator, where there are basically international trials being designed where different sites around the world are doing the identical clinical trial, except that the precise engineering of the CAR is slightly different in one trial to another. And if you do this in several sites, then instead of doing one trial and waiting for the outcome 3 years later, and then doing another trial and waiting for the outcome 3 years later, the goal here is that you have parallel trials going on. And 3 years later, you can pick a winner and say, well, that's the one that really works. So this is really designed to escalate or accelerate the rate at which we will really be bringing CAR T cells into solid tumour treatment for children with cancer. So that's neuroblastoma, which are the nerve tumours, sarcomas, which are the bone or tissue tumours, brain tumours. This is where the field's going now, and it's incredibly exciting, and it's coming fast.
We're running short of time. Have you ever had a eureka moment? Like Ricky Johnson always said, shit, that's the drug, man. When you find something and it all goes down and you've discovered something and you're the first.
My eureka moment has not been so much around a particular drug. It was about an idea. And the idea was we need to be doing personalised medicine for every child in this country with cancer. And there was, as I look back, a eureka moment where I pulled into a room. It was 2013. A bunch of my fellow clinicians and scientists here and said, I think this is what we need to do. And I was confident that we would be able to do it because a couple of those same colleagues and I had developed that minimum residual disease test that I talked to you about, where we became able to predict with pretty good accuracy which children with acute lymphoblastic leukemia, which is the commonest childhood cancer, which of them were destined to relapse. And we did that purely on the basis of molecular genetic rearrangements. And that doubled the survival rate for children with high-risk leukemia over a 10-year period. We started with the kids, 35% of these kids surviving, and 10 years later, 70% of these kids were surviving. And I thought, if we can use molecular diagnostics to double the survival rate for children with leukemia, we've got to be able to apply this to solid tumours. And so I got everyone into a room and I said, this is what I think we need to do. I think we need to harness the country. I think we need to engage government. I think we have to apply for funding to do this. We've got to start the work with the proof of principle here. We have to do this in New South Wales first, and then if we're fortunate enough, we'll roll it out across the country. It was a brave idea, and if I had known then what I knew now about what a huge, enormous, complex beast this was going to be to roll out for the country, I'm not sure that I would have been as brave as I was. But it has been a wonderful story because we piloted zero childhood cancer in New South Wales, in 2 hospitals here at Sydney Children's Hospital, and then in Westmead. We rolled it out across all of New South Wales. And then in 2017, we rolled it out as a national trial. And we were able to publish last year that this is more than double the survival rate of children with these high-risk cancers, irrespective of what their cancer type is, but based on using these comprehensive molecular profiling to identify the best possible treatment for each child based on the genetics and biology of their tumours. So it was more a eureka idea than a
It's still
eureka
a
moment.
Whether it's an idea or kind of need an idea first.
Yeah, and it's been an extraordinary journey because it has required amazing collaboration. It's been very complex to deliver that. It's involved collaboration between clinicians, scientists, data scientists, computational biologists, geneticists. We've had to invent a new workforce, pediatric oncologists who speak molecular genetic changes and computational biologists who speak cancer. We've had to upskill them, we've had to cross-train them, we've had to identify and develop new computational pipelines to analyse all this complex genomic data in the time that it takes to provide a treatment recommendation back to a child within, 6 weeks so that it can impact on their disease. We've had to have partnerships with every paediatric oncology centre around the country to enrol patients and roll it out there. We've now, literally last year, just rolled it out to the first 2 hospitals in New Zealand.
So you can look me in the eye and say, it's working.
Yeah, it is. It's not working for every child yet, so it's not done. We've doubled the survival rates, but there's still way too many children who, as yet, we're not curing. But what we're now working on are the new technological innovations that will, I think, dramatically change our ability to make better, faster, more frequent personalised recommendations. We also need new drugs to target genetic changes that we can see are driving the malignancy in the child, but there aren't any drugs yet to match that particular rearrangement. So we have a drug discovery program. We have an immune oncology program so that we're now able to identify which children are more likely to be susceptible to immunotherapies, and that's another whole area.
Well, I
So,
was talking to Matt Trow, I think. I was in Queensland, and he said, I went to a conference, and it was like, you looked in the conference room 15 years ago with immunologists, 5 people.
yeah, that's right. 2 years ago, I'm a neuroblastoma biologist. That's where my background is. Neuroblastoma is the commonest solid tumour of young children. It's a tumour of the nerves that come off the spinal cord, responsible for about 1 in 6 childhood cancer deaths. Bad disease. Only about 1 in 2 kids with a high-risk disease survive, even today. So with neuroblastoma, but we have a conference, it's every 2 years, the global international meeting. 2 years ago, I reckon there were 2 talks on CAR T-cell therapy. I went in June this year. There were entire sessions, workshops, afternoons, educational sessions on CAR T-cell. You know, in the space of 2 years, a dramatic change in the
It's like
way.
watching the Beatles have a hit, isn't it? Going from nothing to the top of the charts.
Absolutely. So fast, these changes. It's really an exciting journey to be on. It's a ride. It's a roller coaster. You can't be doing the same science today that you were doing 3 years ago, because if you are, you're behind the game. Used to be that if you had a good microscope and you were a good scientist and you had a good head, you could move from one experiment to the next. Now you have to be bringing in new technologies, collaborating globally, using these new approaches, thinking outside the box, just being ahead of those cancers and coming up with new and better ways of attacking them in a way that's safe for the kids. It's amazing. Still amazing to me. I'm going to be 70 next year, and I continue to be exhilarated by the rate of change and the increases in our successes. I look the parents in the eye and the ones who've lost their kids, and they say, I wish it could have happened for our kids. And I say, I wish it could too. And every time I have that conversation, it reminds me that we still have so much more to do, but they're also the ones that inspire you to keep going.
Don't you start tearing up, you'll get me as well. Which means we won't see the next big thing coming, will we? It'll be 2 or 3 years and missed that.
We see them, yeah, sometimes they're here on your doorstep and it's like, wow, I never thought of doing that. And it's fabulous. And then you're scrambling to integrate that into, and that's why, ACRF, why I'm passionate about it, is because they're helping us raise the funds to get these new technologies, to embed them fast, to allow us to adapt and to be at the international forefront. It's fabulous.
You say something nice about them again, but call them the Australian Cancer Research Foundation.
I can say that organisations like the Australian Cancer Research Foundation are absolutely fundamental to our ability to be harnessing, buying, engaging and integrating the latest new technologies and infrastructures into our research so that we can play at the international forefront. We see a new innovation or a new approach we can apply to the Australian Cancer Research Foundation, get the funding, which is often millions of dollars. You can't get it from anywhere else. But if your science is good and you make a good case that if you have this, you can do something unique and remarkable, they will fund you for it. And then it becomes a game changer because you can do things that you could never do before. And if you've got the right team around you, you can do them in a way that no one else can do as well as you can. It is fantastic to have their support.
It's 2:15.
Oh, is it? Then I should be in my next meeting.
I'd like to. I don't think we can go any further with them. I don't want to hold you back. But
That's
thank you
okay.
so much.
Is that
for your
all right?
time.
Yeah.
Yeah, I think it's great. I might call you at another time because I think I'll need a lot of B-roll.
Okay,
Do
that's
you know
fine.
people at the hospital that I can?
Oh, we have our own marketing and fundraising department. They have lots of B-roll.
Oh, okay, but also stuff we might need to specifically shoot as well.
Well, if you need to specifically shoot it, I'll put you in touch with our people and they'll
Fantastic.
facilitate that. That's fine.
That's great.
Okay.
Michelle, you were mentioning before about about, you know, this first drug and it was a breakthrough, but you implied that there were a lot of breakthroughs like that. Could you talk to Stuart about, you know, how many breakthroughs?
I
there were?
can. Just let me check where I am actually meant to be to make sure it's not a meeting that I'm sort of
You're
chairing.
stretching it for us.
No, fair enough. Just,
Calendar. Oh,
Well-
I meant to be chairing
Well, you
the
understand
meeting,
if you've
so
got to go. We don't want to detain.
I probably should go quite soon. But what I could do if it would help, I've prepared, as part of that talk, I've got a couple of 2 slides at the front of my talk, if I could send them through to you.
Yeah,
I don't know
that'd be
whether that
great.
would be
Yeah.
useful. So if you send me a ping, ACRF will give you my details, I'm sure you've got them.
Yeah.
But I can send you a couple of slides that will summarise that, and then if you want me to talk to it, I could do a voiceover or something
Yep,
like that.
fine. It's almost like you've done it before.
Yeah.
Okay, I think we need to let.
I probably should, just because this is